Hello, welcome back to Chatomics. This is Tommy. I have over 10 years experience of computational biology, and today I'm going to explain what is a single cell experiment object in R. So if you work with a single cell RNA sequence data, and especially if you use a bioconductor, you might need to learn what is a single cell experiment object. But first of all, what the heck is even an object? It can be confusing for beginners. So for analogy, for example, you can think your whole body as an object. And you have clothes, you have pants, and you also have pockets in your clothes and in your pants. And you can also have keys, for example, in your pockets in your clothes. So you may have 1 key, 2 keys, and 3 keys. And you may also have like a cell phone, for example, in the pocket of your pants. Okay, you can think single cell RNA-seq experiment as an object, right? And it has different pockets in this object, for example. So different pockets here, like in our term, is called slots, but you just think them as pockets. For example, different pockets can contain different small objects, for example, the keys, the phones, right? So in here, a single cell experiment objects, then the main pockets here is called assay pockets or the assay slots. So for example, it contains, for example, the raw counts. So in this case, each row will be 1 gene and each column will be 1 cell. So it can have 1 assay called raw counts, right? But you can also do normalize log counts for the same actual count matrix. So that's why you can have multiple different assays in the assays slots. Okay, and then moreover, it has this called row data slot. So this contains information for each row, essentially each gene in this case, right? So for example, you can have more information for each row is 1 gene, then you can have, for example, different gene IDs, ensemble ID for that gene, and maybe official gene name for that gene, and even the length of that gene. Even it can contain the ranges of that gene, for example, the chromosome and start and end for that gene. So moreover, you also have this called call data or column data. It should contain the cell metadata. So each row here will be 1 cell. So corresponding to here, each column, okay? But here will be each row here will be 1 cell. Then each column here can contain different metadata. For example, different batches, different maybe cell annotations. Each row will be 1 cell. Like maybe that cell is CD4 T cells or CD8 T cells, right? Okay, so that's actually the main slots here. But for single cell RNA sequence data, there's this slot called dimension reduction or reduced themes slot. It contains the dimension reductions. Okay, so each row here is the same. It's 1 cell, and each column here, for example, if it's a principal components dimension reduction PCA, then each column will be 1 principal component, PC1, PC2, PC3, right? And if you work on single cell RNA sequence data, you know there could be other dimension reduction, nonlinear dimension reductions, for example, t-SNE or UMAP. So that's why you can have multiple dimension reductions in this slot, okay? So in the following video, I'm going to demonstrate how you can construct a single cell experiment object from a count matrix download from GEO, and how to actually deal with all those slots actually in R. Okay. So the data we're going to use is, for example, is this data sets from GEO GSC 126030, and it contains I think different cells, like some of them are PBMCs and some of them from human lungs. Those are human normal tissues, and they compare the the T cells actually in the resting states versus the activation states using the CD3 and CD28 activation. Okay, so this is the GEO. So I will have all the scripts and also the links in the description of the video, so you can follow this if you really want to. Okay, so you can actually directly just click here and download the count matrix for all the samples. But I usually go to the FTP site from GEO, and that's actually how you find that this raw tar file, the compressed file here. So here is the FTP address, and then this is GSE. So you specify the first 3 digits, and then the last three digits use NNN. And then specify the whole accession ID and the supplementary. Okay, so if you go back, okay, so if you actually remove the supplementary, it actually contain all the files here, but it will be in this folder. So what you can do, okay, let's actually go back. So you can actually right click here and then copy the link address. Okay. So now let's go back to RStudio and download that file. Okay, so here, okay, for example, you first need to actually install a bioconductor and also the SingleCellExperiment package and other packages that we will use. For example, one of them called Skater, and load the library. I already have them installed, so I'm not going to run those lines here. And this is the URL I just copied. Okay, so I will go to the terminal and say wget, and this is the URL for that tar file, the compressed file. So if I do that, it will be downloaded. So if I do ls on this thing, so it will be this file. So you use this tar command to decompress the files. Okay, what's going on here? Oh, sorry, there's a different name. Okay, so copy that and paste that. So if you do ls again, then you see those count metrics. Okay, and you see each actually count metrics here for one sample. Okay, we'll just read in one sample and use that as an example for demonstrating single cell experiment object. Okay, so but first of all, we need to look at the file and see how it looks like. Okay, so if I example let me go to the data I downloaded here. So just look at one of the files. Okay, I used, because it's a zip file, I used zless. You look at that file, so each row will be one gene, each column will be one cell. But something actually is special for this data set is that you have actually two columns here. Like one is the ensemble ID, the other one is the official gene symbol. So you need to probably take care of that. So every data set is a little bit different. So you really need to look at data and see how you can make them into the right format for your analysis. So let's see. So let's read that into R. Okay. So library tidyverse library here. So I'm going to just read in the second sample. Okay. If I do that, okay. So be careful actually when you need to pay attention to all these messages from, for example, from those packages. Then you see there's something called, oh, there's a new name. This thing converted to whatever 2466. Okay, and you say, okay, there's some warnings here, and for example, you can look at the problems of that data. So although we read into R, but looks like there's some problems. They said, okay, you have actually 2,466 columns are expected, but actually we only have 2,465 columns. So what's going on here? So we can look at the column names of that data frame and then look at the last 10. And you see, okay, those are all actually cells, barcode, but the last one is 2466. Okay, that's actually something we saw here. They have these new names. But let's look at that last column. Okay, and look at the first 10 rows. Okay, just use $sign to access the last column, and then look at the first 10. Those are NAs. Okay, and then if we just look at how many NAs in the last column, you see they're all NAs. So the problem here is most likely when the authors upload the data, the last column is empty actually column, but somehow it gets read into R because for example, in Excel, like you have an additional column here, and without filling anything, but they still get read into R, and it's empty here. So let's just remove that last column here. So total we have 2,466 columns. Let's remove that last column, and now if we look at, okay, now you have the first 5 rows and 5 columns here. Okay, let's convert that into a matrix. Okay, so remove the first 2 columns, use this syntax, and then pipe it to a dot matrix, so convert it to a matrix. So this was data frame, so let's convert it into a matrix. Okay, and this matrix, we can look at the first 5 rows and 5 columns, and it's like this. And then we need to actually add the row names as the gene name here. So there's no gene names here. So let's do that. Let's look at it again. So now it's a count matrix with row names and column names. The rows are genes, columns are cells. Okay. Okay. Once we have the count matrix, we're ready to create a single cell experiment object. Okay. So in R, it's very easy because you just use this construction function called single cell experiment and specify the assays. So assay, you remember, you can have multiple different assays. It's a list. So you can specify the counts, the name of that assay, and this is the count matrix we just created here. So let's do that. And now you have a single cell experiment object. So if you look at that, you'll say, okay, this is a class of single cell experiment. This is the number of rows and number of columns here. Okay, and then you see in assays you have 1 counts and the row names, the row data, and which we don't have anything, and the column data we also don't have anything. So reduced dimension, and we don't have anything either. Okay, so to access the count data we just supplied, we can do one of the following using the getter function. So for example, assay, specify the single cell experiment object, the variable, and the counts. Okay, this one. Let's do that. Actually, we'll return that whole count matrix, but we can just look at the first 5 rows and 5 columns here. Okay, like that. And you can also use this convenient getter function called count SCE to get the same results. Okay, so those two are the same, but this is more actually general because you can have multiple different assays, and you can always use this. For example, you have a log count, so you can see assay parentheses SCE and log counts. Okay, to get that log counts matrix. Okay, so you can actually access the slots. Okay, whatever the pockets using the at operator. So this is, however, this is considered as a bad practice, as the class developers are free to alter the internal structure of the class and maybe that at sign will not actually work. So rather always use the getter function like assay and counts. Okay. For example, just to demonstrate, you can look at the assay pockets or the assay slots using the @. If you do that, and you say, okay, it's an object of the assays. It's an object of class simple assays. Okay, and it has that object has a slot called data. Okay, so you can use the @ again to access the data. Then you see the data is a list of length ones is a count. So if it's a list, you can use the $ to access that list, the elements, and then look at the first 5 rows and 5 columns. And you see this is actually essentially the same as here, right? But this way to access the assays is the recommended way. Okay. So we can add actually more assays. For example, we can do log normalized counts. So if you use a different actual package to calculate the log normalized counts, and will put into that single cell experiment. For example, if you do that, okay, it takes a couple of seconds. Okay, so if you look at SCE, and in here now you see the assay slot, you have now two different assays here, like counts and log counts here. Okay, so for example, now you can access that by SCE @ data, and because it's a list of 2, so you can access the log counts by using this. But for example, you can also use assay using the get function SCE log counts, and then look at the first 5 rows and 5 columns here, and get you the same result. Okay. So you also have this like convenient accessor function, getter function called log counts, and actually will get you the same actually like here. But you can look at the dimension, for example. Okay. So you can also add those assays manually. Okay. So for example, I'm creating count matrix using the original counts and just +1 add this pseudo counts 1, and then if you do that. And you can use this syntax called assay SCE, and the name of that, okay, assay counts underscore. You can specify anything here that is sensible to you, and then put assign this new counts matrix to the assay. For example, you do that, and now if you use assays, for example, now it's plural. You see that, and you see it's a list of length 3. Okay, now you have counts, log counts, and counts +1. Okay. Of course, you can add this new assay data like this. For example, you can use the @ to access that slot, and then use @ to access the data slot. And now because it's a list, so you can just create a new element. For example, counts 2, and then you can add the log counts +2, for example. Just there's a dummy example here. And if you do that, if you look at it now, it's a list of length 4 counts, log counts, counts underscore 1, and counts 2. If you do this, okay. And of course, you can use this assay names function, and it will show you all the names. Okay. Okay, now we can actually add some metadata for each cell, so each row. Okay, you use this Scala package to add the QCs for each cell. And now you have the column data. Okay, if you use the call data function, you do this, and it returns this data frame. Actually, now each row will be 1 gene, each column will be some metadata for each cell, for example, the size factor for normalization, and the number of total UMI counts, or how many, for example, how many cells, how many, for example, detected genes, for example, here, okay? So you can, for example, you can save that into a new variable called data, and if you print it out, it's just this data frame, right? And the row names of this data frame should be the same as the column names of the original count matrix, right? Because the original count matrix, each row will be one gene, each column will be one cell, right? And here each row will be one cell, and you want to make sure the order of those cells are the same, okay? So if you use identical here, row names of that column data and then column names of that original count matrix, they are the same, okay? True, okay? Okay, now we have the column data, and like we also can add the row data. Row data, essentially each row will be one gene, so you can, for example, you can add use this function add per feature QC, and then if you look now, if you use the row data getter function SCE single cell experiment, then now you also return a data frame. So now for this data frame, because it's row data, each row will be one gene, and each column will be some data information for that gene. For example, the mean expression level, or the, here maybe is the detected, for example, percentage, for example, for that cell, for all the cells, for example, here. And you can even add, for example, the gene length or the genomic coordinates, chromosomes start and end, using the row ranges. So now it's just empty here, so it's a genomic range list object. But now it's empty, but you can actually add those informations too. Okay. So in my next video, I'm going to explain the thread object, which is the different implementation of the single cell object, and I will say the column data is more like the cell metadata. It's very similar to the metadata slots actually in thread object. It can be actually confusing because in single cell experiment you also have this metadata slot, which is different from actually the metadata slot from thread. And here you can actually add any arbitrary named list of arbitrary objects into this metadata slots. For example, my favorite genes, or maybe the most variable genes, you can actually add that into the metadata slots. You can use the metadata, and it's a list. And if you use the the getter function, you see it's a list, okay, named list. Okay, finally, that's a very important thing for single cell RNA-seq is the dimension reduction slot, okay. So let's actually calculate the log normalized counts again, and then we can run a principal component analysis for this count matrix, log normalized count matrix. We do that. Okay, so run PCA. If you look at the single cell experiment object now, previously it was actually nothing here, right? The reduced dimension. So now you have a PCA here. So you can actually use this getter function called reduced dim, and then the object name SCE and the PCA. And you look at the first 5 rows and 5 columns here, okay? Again, each row is one gene, each column will be one principal component, PC1, 2, 3, 4, 5, okay? And you can, for example, add another dimension reduction into that slot, because, for example, you can run SNE, and if you look at, so see here is the plural reduced dimensions, SCE. This is a singular form here, okay? So if you look at this, and you tell you it's a list of 2. Now you have the PCA and TSNE, okay? But you can actually access that. Let's see, for example, yeah, you can just use this thing to access the TSNE coordinates. Again, the rows are cells, columns like TSNE1, TSNE2. And similarly, you can add UMAP. Okay, each row will be one cell, each column will be like UMAP1, UMAP2, for example. Okay. So yeah, I will skip that. And I think that's probably the majority things that you need to understand for dealing with a single cell experiment object. So I hope it's useful for you, and click subscribe if you like this content. I will see you next time. I will explain what is a thread object in R. Happy learning.